Our next presenter is Anusuya Willis, and I haven't got my piece of paper. But I know Sui is going to talk about the National Culture Collection. Sui works for CSIRO, and she's going to tell us a little bit about the strains that they have in the National Culture Collection. And her talk is actually titled Cyanobacterial Strain Variation, Implications for Research. And joining her on stage is her little son, Jasper.
Cool. Thank you. Thank you very much. I'm very happy to be here today. And the last talk of today is about cyanobacteria strain variation, which we have already heard quite a bit about today. So I'm going to talk a little bit about what is strain variation. We heard a nice talk from Sandra this morning talking about species variation and how variable they are. With strain variation, we're talking about the variation within a species, so different strains of the same species and how much they can vary. They can vary in morphology, of course, and here's an example of Ophidiopsis racebuskii, previously known as Cylindrospermopsis. And you can see immediately here that there are coiled ones and there are straight ones. So this is an obvious way that they can vary. They can also vary in their physiology. And in this example here, there are 24 strains grown under the same conditions, and this is their growth rates. And you can see they vary about, they double, basically. There's a big variation there. And their toxin cell quota also can vary quite a lot, by about threefold. And these are the same strains, different cell order. But you can imagine that a population of this strain, SO9, will have a much lower risk to the water body than a population of, say, SO3, which has 3 times the amount of toxin production. These strains also have a genetic basis. So the important thing about these 24 strains that I just showed you is that they all come from the same sample and the same water body. So these strains are all living together. And there is a genetic basis for these differences. These strains have about a 15% variable genome. The core genome is about 85%, and their genome size is 3.8 million base pairs, which is very small, and small for Anostopali cyanobacteria as well. We can group strains together based on their physiology, and we did this with these 24 strains here using a Euclidean distance plot. And you can see that the strains do base together based on their traits. Here we have them based on a high cylindrospermopsin to deoxin cylindrospermopsin ratio. Some group together based on a high cell quota. Others a high growth rate. Also the straight and the coiled group separately as well. And these groupings show their ecophysiology, so basically their micro-niche separation. So even though they've all come from the same environment at the same time, their optimal environmental conditions are slightly different for each strain. So how much does this matter? Phil talked about this earlier. We've also heard other talks about strain variation. And in this graph here, we can see samples taken about every 2 weeks over a summer bloom of Cylindrospermopsis. And you can see that the amount, this is the average toxin cell quota, you can see that it changes quite a lot over these 2 weeks. So this has an immediate effect on the quality of the bloom and why strain variation is important. Strain variation is also really important for research purposes, particularly in competition experiments. And this graph here comes from Man Xiao's PhD. And it is a modelling experiment where we see the outcome of 2 strains competing together. The yellow squares show 100% win by a single strain. The blue squares show a 50-50 where there is no win, or they coexist. And you can see that there is a big variation in the results. There isn't a single dominant species or strain between all these strains. So we're getting a lot of variability. So if you were doing a competition experiment and you were using this strain, you would feel like you had a very dominant strain on your hands. But it really depends who you are competing it with. If it competes down here with this strain, then you get a completely different result. This is very exciting. This brings me to the second part of my talk and my new role at the Australian National Algae Culture Collection. And how we are keeping cyanobacteria strains to promote research. We have 350 curated strains of cyanobacteria in the culture collection. Only 168 of these are currently available because this includes species that are dead, basically. The 168 cultures encompass 17 species. And for example, we have 38 strains of Microcystis, 15 strains of Rhaphidiopsis, 26 strains of Dolichospermum, and 53 strains of Nodularia. And what this looks like, if we look at Microcystis, we have strains collected from about 14 different locations across the southeast of Australia. For Rhaphidiopsis, we have 15 strains collected from only 5 locations. For Dolichospermum, they are collected around Australia as well. In the culture collection, we are sequencing the barcodes of everything in the collection, including the cyanobacteria. We have more than 1,000 strains, so this is taking a little while, but we're making our way through. We maintain research lab rats, like CS505, which is Rhaphidiopsis, which I've used a lot for my previous research. And we are looking to expand and update our culture collection with new and emerging strains, particularly problematic strains. You may have noticed that all these strains that we have in the collection were collected in the 90s, so that's quite a long time ago. What would be really great to have is some of the new problematic strains. Nijoy made it very clear that it would be wonderful for us if we had had a sample from the Murray River bloom 2 years ago. But in the culture collection, how many strains should we have? Because we are constrained by time and space with how many we can keep. And how can we best support research? How can we best support everyone in the community who would like to use these strains? So these 24 strains collected from a single sample is obviously too many for us to maintain in the culture collection as living strains. But if we look at the Euclidean distance plot and go back to a Euclidean distance of 3, we can group these strains based on their physiology, and we can group them into, in this case, 5 groups. And the genetic basis for these is about 15% genome variability. If we go in a little bit and a little bit less conservative, we can get a grouping of 10 strains. And some of these strains within the groups only have 1 or 2% genome difference. So for the sanity of us at the culture collection, we'd probably go for the 5, and from one sample, look for 5 strains, even if it requires quite a lot of preliminary work to understand the diversity within the system or the sample. So the collection at ANAC, we would like to have a collection that showcases the Australian diversity of the species and the strain diversity, which includes spatial and temporal diversity, and of course multiple strains from single locations. We'd like to have strains that came from environmentally important locations, new blooms, emerging blooms, shifts in the dominant species, such as the Murray River, and also economically important sites, say persistent blooms in reservoirs. But for this, we need your help. So we can accept, maintain, and curate new isolates and strains. We can do isolations ourselves as well, and we would like to increase the cyanobacteria collection. And that brings me to the end of my presentation. I'd like to thank my current co-workers, Ian Jamieson, the director of ANAC, Catherine Rossi, who maintain the cultures, and of course my previous colleagues, Michelle Burford, Anne Chung, Manjao, Phil Orr, Jason Woodhouse, and Brett Nyland, who I did all the strain variation work with. Thank you.
Thanks, Sue and Jasper. Do we have any questions for Sue, please? Michelle.
I didn't want to ask a question because
You know everything.
yeah, so though, as you know as well as I, those particular strains that were done in the toxicon study were all from a bottle of water.
Yes.
How do you reconcile strain variation at a small scale versus a lake scale versus a regional scale versus a country scale?
Yes. So at the moment, we don't have enough information about that variability between strains to be able to make a decision, particularly in the culture collection. We would need to do some morphological studies to see how much the variation changes, make these Euclidean distance plots or other trees, and then choose the strains based on that information. But right now I can't say. But we can start. Yeah.
Anybody else? Well, I think you've told them everything that they need to know. All right, ladies and gentlemen, thank you. That's the end of this session. But I'm actually going to put Michelle Burford on the spot. Michelle, would you like to come up here? Just so we've got a few minutes to spare.
What, karaoke or something?
Yeah, we'll do a duet. I believe you know something about the Twitter Cyanobacteria conference that Manzhou is organising, and I believe that at some stage you were going to mention it.
Yes, I was, and I actually had it tagged on the end of my talk tomorrow, but I'm happy to talk...
Okay.
No, I'm happy to talk about it now because that means I get more time to talk about other stuff tomorrow, right? And Sui has very nicely provided a segue for that by flagging this with her little Twitter. The fact that on the 8th of... at 8 a.m. it's going to start, 8 till 4 Australian Eastern Standard Time, which is going to...
Oh, that's 8 a.m. France. That's what I'm...
Yeah, this is going to be interesting. It's going to need a bit of clarification because, in fact, it'll be daylight saving in New South Wales. Yeah, I'll get to that. Daylight saving in New South Wales, but it won't be in Queensland. So Mandy may not have thought of that. Anyway, it's a global Twitter conference. Now, I've never been involved in a Twitter conference before, and it's going to be kind of an interesting thing. But the topic, it's on the 24th of October. The topic is cyanobacterial blooms under climate change. And there will be a series of presentations, of course not as we know it, but in the Twitter form, where people will get sort of 6 bytes of tweeting along with associated graphics or photos or whatever about a particular topic. For example, Sui's giving one. There's a range of Jonathan Puddick's giving one. There's a range of people around the world who've already signed up to be involved, and it's supposed to be completely interactive. So people tweeting as we go, responding. So it kind of be interesting to see. People are going to have to obviously give really short, sharp presentations about things to do with cyanobacteria, and people will be responding around the world. Now, obviously, it's nice that we're in charge here for a change and we're not at the mercy of the European or the American time zones. And so the Americans will probably sign in early in the day, whereas the Europeans will sign in late in the day, and we'll have all the middle of the day, right, for presentations. So I'd encourage people to consider being involved. It's kind of going to be an interesting trial to see how it goes, but it is the opportunity for some genuine dialogue, maybe in a way that some people feel more comfortable with than asking a question at a conference or something like that, to really throw in some comments and ideas and challenge people's presentations. I guess hopefully all the information, well, I will put up all the information tomorrow anyway, if you want more details about it at the end of my talk. And come and chat with me if you want more information. Or you want to start on Twitter for the first time. In fact, I just tweeted Sui and Jasper giving a talk. Thanks, everyone.
Okay, thanks, Michelle, and sorry for putting you on the spot there. There is a little PowerPoint slide that I've printed out, and I'll pin a couple of these up around maybe at dinner or whatever, and I have some more detailed information in my bag. So if you are interested, there's the information, and the more detailed information I'll put up as well. There is a website you can go to, and that's on there as well. So so the Cyno TC 2018 Twitter conference is going to happen.